wait 5 more minutes probably.
Yeah.
Yeah. Oops.
Okay, it works. And
Yeah, I can see the live stream on YouTube.
Andrada, did you try the microphone again? I don't remember.
Yes, can you hear me?
Yes.
Yeah. Oh, perfect. Strong and loud. Excellent.
I guess I'll send a last minute email to Joao just in case he mixed up the time zone.
Yeah. I will text
Yeah.
him. Oh,
Maybe.
he texted me. He says he has a hard time logging in the Zoom link.
Okay.
Does he need to be registered?
He can put so if when asked for the email joining the webinar, if he puts like the personal email, he shouldn't have this issue.
He should be there.
Ah, I see him. Yeah.
Hello, Joao. Welcome.
Hi. Sorry for this mess. Was a bit difficult to log in somehow. I don't know why.
No worries. We had other participants that had kind of conflicts between different institution emails, so no worries.
Perfect.
Do you want to rapidly try to share your screen so we are all set?
Yeah. Can you guys see it?
Yeah.
All right.
Yes.
Let me just see if in presentation mode it works fine. Yeah, works fine, right?
Yeah.
Just...
So since it's
go.
here, I guess that I'll start record, and Chantal, whenever you want, you can introduce the speakers.
Yeah. All right, let's start. Welcome everyone back to the BrainHack Micro2Macro. We will start today with the unconference with 4 excellent speakers. We're absolutely thrilled to have you here. And the title is Finding the Needle in the Haystack, a Hardcore MRI for Microstructure. Please, if you have questions, you can post them already in the Q&A during the talk. If you're following this on YouTube, you can also post questions there or post them in the chat so that we can make this as interactive as possible. And the first 2 speakers will be Gonzalo Alvarez and Analia Zwick from the Laboratory of Nuclear Magnetic Resonance Spectroscopy and Imaging in Argentina. And they will talk to us about quantum physics for deep tissue magnetic resonance imaging.
Okay.
Well, thank you very much. We will share now the screen, and
Well, thank you very much to the organizer, first of all, to invite us to this conference, to all one that is there connected also. And I think this is one great thing of this pandemic time that allow us to have more this online scientific meeting and to connect each other despite the place where we are. So thank you very much. And well, as Chantal say, or Chantal say, I don't know how to pronounce it. We belong from the laboratory of NMR and MRI from a new medical physics department in the Bariloche Atomic Center, which is located here in the south of Argentina. That's why we are in summertime now. And in Bariloche, which is surrounding of this amazing views and nature here. And we are running the lab together with Gonzalo Alvarez, who is here with me and who is the expert in the NMR and MRI techniques. We both are physicists, trained more in quantum physics, and we use that in our laboratory to exploit fundamental quantum physics for developing novel applications and technologies in physics, biology, and medicine. And in our lab, we have this equipment here. This is an NMR MRI magnet, which is the first one in Latin America with high field, allowing to produce images for in vivo small animals. And well, which are the kind of things that entertain us and the challenge that we propose us in the laboratory, where one worldwide challenge that exists is for fighting diseases like such as cancer and neurodegenerative disease is to understand at the cellular level the biological and biophysical process that produce them. So, and nowadays the MRI imaging, that is a great technique because it's non-invasive, have about the resolution of a cubic millimeter, which is great for several purposes, as we know, but it's not enough to distinguish or to recognize those disease at early stage. And also it's not enough this resolution to see all the connection that we have inside the brain, although the tractographies are great tools, still are facing huge challenge to recognize all the paths that we have inside, because the actual resolution of the MRI images cannot distinguish, for example, the crossing of the fibers that we have inside the brain. So, and even less to distinguish what is happening inside of all of these fibers, that we have a universe of things and microstructure there. As we can see here, this image here below is, we cut one slide of this cable connection here. We can see here, and we put inside of a microscope. What we have here is a lot of axons, and each axons, the diameter of them range between 1 to 10 microns. And for example, it's believed that one early indication of the Alzheimer's disease is to distinguish damage in the myelin layer that are covering all those axons, that it is this black layer line here. And this, as you can see here in this scale, is around 1 micron. So we are talking about to distinguish something that is about 1,000 less smaller than the actual resolution of the MRI images. So we ask here, so how to break this barrier and to be able to see in a non-invasive way the universe of cells and microstructures and processes that are hidden between each millimeter pixel of a resonant images. So to answer that we use concepts from fundamental quantum physics to break down those barriers imposed by the current technologies. And the key point that we used to do that is that inside of all these cavities, is happening the transfer of different substances, including the protons of water molecules that have quantum properties like the spin. The spin is something that is very sensitive to nuclear magnetic resonance, and is because of that with NMR techniques we can monitor and we can interact with them and extract some information from inside the body. So, and we are full of water in our body, so we have 75% of water, so we have a lot of those spins, and we can use them as sensor, like a spin sensor that can monitor. If we mix the quantum information processing tool with the control tools of the NMR, we can design quantum sensor to probe the matter and what the process that is happening at this micro scale. So we use the quantum sensor sensing, which is one of the new quantum technologies that contribute to develop, to pave the way to develop also non-invasive biological medical applications using NMR and MRI techniques. And with those, we try to develop tools for virtual histology to identify biomarkers of those disease in an early stage, and to be able to produce with the kind of information that extract from this quantum sensing, to produce quantitative images of relevant parameters that in a micro scale. So, like the one that we are showing here, that where Gonzalo will explain much more in detail now in the following.
Okay, thank you very much, Annie. And so I will tell you a bit, so this is the motivation mainly of our research line, so I will tell you a bit what are the tricks and how we can encode all this quantitative information in the images. So essentially, as Annie mentioned, biological tissues are mostly water, so we have a lot of molecules of water. And what happens with the molecules of water is that they suffer a process that is called diffusion and moving randomly within our body or our tissues. And in some sense, they are a spice of the microstructure of our tissue inside, because they are moving within our tissue. So just to give you a picture of how this process is happening, imagine this is a glass, is some kind of simple model of a tissue microstructure property. So this is it can be like a very simple representation, for example, of a cell. So we have water inside of the glass, and imagine we put a drop of tint inside of the glass of water. This will show how the water molecules are moving inside of the glass. So this is what is shown in this picture. And what you will see at the beginning, this water will start to move freely. And if it's in the center of the glass, we'll not see the boundaries of the glass. So the diffusion process will be something like, it will be free. It doesn't have information of the boundaries. But after some time, the tint will start to explore all the glass. And in some moment, this water molecule will start to hit the boundaries, and will start to change the way that they are moving because of the boundaries. So depending now of the time scale that we look at this molecular diffusion process, we can have information of something that is called free diffusion, because the molecules can move freely, and eventually in some time they will start to see the boundaries, and the diffusion process will be different, and in some sense the water molecules start to have information about the tissue microstructure. So what is important that in NMR, we work in a strong magnetic field. We put the sample, let's say a brain, inside of a magnetic field. It's a strong magnetic field that is static and homogeneous. And what happens is that the nuclear spin that are inside of the water molecules presses around the magnetic field with a given frequency that depends on the strength of the magnetic field. So what we do is to apply what is called a magnetic field gradient. So in such a way that the magnetic field now is inhomogeneous, and depending on the position of our tissue, the magnetic field that the spin will see will be different. So we'll precess with a different frequency, And what we do is to look at the precession frequency to distinguish what is the position of these molecules. So with that, the NMR technique is very sensitive to distinguish the movement of the molecules. And this is something that is called in NMR diffusion MRI, and we can detect we can detect the movement of the molecules, the liquid molecules. So this is the way that this is the physical process that we will use to try to extract quantitative information to try to solve the problem that Danny mentioned. So this is an invasive image, and what we want to try to do is to try to extract this information, let's say of the diameters of cells or axon or whatever property of the tissue microstructure. We want to use this diffusion process to extract this quantitative information. Just to give you an idea, the diffusion MRI is a technique that is used every day. It's a clinical practice also that is used. This is, for example, an example of a technique that is used to do some kind of diagnostic. So this is a person that have an infarct in the brain. The left figure is a conventional MRI image that doesn't show, you look at the brain, it looks symmetric, so it doesn't seem to show a damage, but this person have an infarct, and if you look at the right figures, they are based on looking at the diffusion process of molecules, of water mainly, and if you see there is a bright part, a very white part, that is showing that the molecules are diffusing more slowly than in a normal brain, and this is some kind of a witness of the damage that was done in the brain. So this is some kind of technique that you can look at the qualitative changes of the diffusion process to make some kind of diagnostic of the damage that the person suffered in the brain. But the point is that most of the used techniques essentially are a qualitative image that show changes in the diffusion process. So now the people, there are a lot of scientists that are trying to have more precision and to generate quantitative images and start to characterize with more specificity the tissue microstructure. And this is what we are essentially working on. And this is what we use a quantum information theory. So because our body is built by quantum systems, so we are using theory of quantum information to try to encode quantitative information in the signal of the NMR that we will detect. And essentially what we do is we need to characterize something to extract quantitative information. So it's very important to find relevant parameters that describe our tissue microstructure. So for example, if we are looking at axons, something that will be very nice, it will be, for example, to measure the diameters of the axons and try to make an image with the information of the diameter of the axon that we have in different pixels of our brain. So essentially, this is what we are trying to do, is to identify parameters and encode it in the NMR signal, and with this parameter then to make an image that is quantitative on some parameter and make a map where here the pixels are very large. They are about 100 microns by 100 microns. But what, for example, in this image, what we did is to look at the average restriction size of the diffusion process and put it in the image. So the different colors and giving information about the mean size of the restriction sizes that the molecules are suffering. And if you look, you can see the color scale is showing sizes of the order of the micron. That is much lower than the resolution of the MRI. And we can also detect very small changes. So essentially what we do is to use these quantum information theory tools, quantum control tools to design NMR and MRI pulse sequences. And also we use some tool of real-time adaptive estimation to try so the goal is to try to extract this relevant information efficiently in time and precision, so we can do quantitative images, and it's very important to do it in a short time, so it can be practical for clinical studies or in vivo imaging. So this is, for example, this is an example, as I told you before. Here, essentially, we put the mean size of restriction sizes, but tissue microstructure is very complex and is very inhomogeneous. So if you look at this image, we have different axons that have different shape and different diameters. So the information is not only one parameter. We have to try to get information of more details that characterize this tissue microstructure. So for example, this can be a size distribution probability of how the diameters within the pixel are represented by a size distribution, for example. So what we do here is this is an experiment with the ex vivo mouse brain. We are focusing on the corpus callosum. Here I'm showing some region of interest within the corpus callosum. And we designed some techniques that are very sensitive now to extract information about the size distribution of restriction lengths that are inside this part of the tissue. So in the middle plot, you can see the size distribution that we extracted. And if you look at the different region of interest, the size distribution are very similar. To the eye, it's very difficult to say what is the difference between them. But if this very small difference is enough to distinguish different region of our brain. So in the left side, what we do is to use the size distribution and, for example, to show in this particular figure the mean size of each of the distribution. And you can see that in the color code map, you can distinguish 5 regions. That these 5 regions were identified by invasive studies. If you do histology and you measure the diameter of axon in these different regions, the people saw that there are very subtle changes in the sizes of the diameter of the axon in these different regions that are very small, but are different. And in this picture, we were able to distinguish these regions. And if you see in the map, we are distinguishing sizes of the order of the micron, and the changes in the regions are of the order of the nanometer. So this is showing the potential of this technique if you are able to extract quantitative information, how allow you to distinguish very subtle change in the tissue microstructure. But so another problem that have NMR is this technique is that we still don't know how the tissue microstructure properties are reflected in NMR signal. So what we are doing essentially is to model, to make some hypothesis of a biophysical model that describes the tissue microstructure to try to extract quantitative information. But we still don't know, it's very difficult to design proper models that describe, for example, size distribution of axons or some property of this tissue microstructure. So it's very important to design tools that you don't need a prior assumption of what is the model that describes the tissue microstructure. So this is something that we are also working, and recently also we published a work with a tool that we design a method that we call it size filters. We designed NMR techniques that can only see the signal of spins that are within a specific size in the tissue microstructure. So this is, for example, some kind of filter that we change some parameter of the NMR sequence. In this case, is the gradient strength. And depending on the gradient strength, we can choose what size we can see in the tissue microstructure. So in this top panel, I'm showing again an image of a corpus callosum. And as I show you here in the splenium, we have a region that the sizes are very small. They were seen that they are very small. So if we choose a proper gradient, we can highlight only this region that is the splenium. The red is more signal, and we can highlight the region where we have larger size of restriction lengths. But then if we change the parameter, for example, here is the isthmus, and there we have sizes in the axon that are larger. And if we change the parameter, we can highlight the region where we have larger sizes of restriction lengths. And essentially with this filter, what we can do is to scan the size distribution so we can produce a model without assuming the model from the beginning. So more or less, this is a picture of what we are working on and how we are mixing quantum information tools to extract information and produce quantitative images. And we are also doing, so we are doing that on a preclinical experiment with the ex vivo tissue, but we are also working with colleagues that have access to clinical magnets. And we are now working on trying to implement this kind of sequence also for study human brain. And eventually we hope that in some moment we can use it also for in vivo with humans. So with that, we want to thank you again for the invitation and our collaborators and our group. And thank you very much for your attention.
Thank you very much for that presentation. It's great to see a quantum perspective to this, and that's what makes this field also so interesting, because it's so multidisciplinary. So thank you so much for talking about your perspectives on this matter. If there's any questions from the audience, you can post them in the Q&A or on YouTube. In the meantime, I have a question. You showed these size estimates, and there are a few techniques developed that do this. For example, the Excalibur model or power law approaches. And I was just wondering if you could comment on how your approach compares to these approaches.
Yes, so we didn't do yet a comparison to try to find what is more, I mean, there are different problems. One is to make a technique that is practical, and you have to extract this information in a fast way. And other thing is to try to have a proper information. So what we are doing, the diffusion process is essentially, the technique is based on diffusion MRI. So essentially what we are doing is, what they say in some sense, that we are using these quantum information tools to try to find how to design the method or the protocol to extract this quantitative information in a fast way. And the problem is that there are no, in general, there are no unique way. There are no only one optimal way. So it's what we are getting results that, depending of the size or depending what is the tissue microstructure that you want to estimate, this will tell you what is the optimal way to extract it. So we are using these quantum information tools, first of all, to find what is the optimal sequence to extract this quantitative information according to the diameter or the size that you want to estimate. And the next step that we also do some theoretical work, we want to do some kind of online learning, because sometimes if you don't know exactly what you want to estimate, first you have to make a first estimation, and with that information you can redesign the technique to do this optimization. So essentially the main difference that we are exploiting is these quantum information tools that there are some kind of metrics that tell you what is the optimal way to extract this quantitative information.
Because the optimal parameters to set the sequences depend on the size that you want to estimate, but you still don't know them. So it's like, because of that, we implement this kind of adaptive methods that you can do something, some measure, and from this learning and different, and to modify the sequences to get the information, the right information, and to converse that.
That's very interesting. Yeah, thank you. I'm just looking if there's any further questions. Matteo, are there any questions on the YouTube channel?
No, so far no question.
I think we have Marco with the hands.
Oh yeah, Marco.
Yeah, thanks for the great talk. I was just curious to follow up on this sequence optimization. So you have shown an example where you modulate the gradient as a filter, but I presume you also account for the timings, right? The diffusion time, for example. So is that accounted for? And,
yes. So the way to do the filter depends on all the parameters of the sequence, as you want to. All the parameters of the sequence. This can be the diffusion time, the gradient strength. There are different parameters that you have to choose them in such a way to design the filter to see what size you want to measure. but
And
it's
what-
a combination of them.
And then you have different limitations. We also studied that you have different relaxation times that avoid you to go to long times. So it depends on the tissue that they are trying to look at and to get some information. And also the gradient strength that you have are different depending on the equipment. So that is a limitation that they will at least allow us to say, well, with this kind of equipment, we can do this and not go beyond that. And we can know which are the limitations of the equipment and what we can implement there.
Yeah, about the confounding additional effects, relaxation is one, but maybe also permeability. So
Yes,
you have a way to account for exchange, because, okay, for axons, healthy axons, we can kind of assume long exchange, but
yeah,
at the moment, I mean, first of all, what we do is to design a method that is based on a restriction that there are no permeability or something like this. So it's a filter to distinguish sizes. Of course, one can assume that you have permeability and the model will be different. So in this case, we have to you have to design a different method. But
this is one of the follow-up steps.
Yes. So at the moment we are. I mean, it's too complex the tissue microstructure.
Yeah.
So to have a specificity is very difficult to all the parameters. So you have to focus depending on the problem to try to find what is the best way to solve this problem. But I mean, you have to do it step by step. So we started. We are starting from for the most simple things that are more connected also with what is done, and we are starting to add additional parameters and see how we can distinguish the different physical processes.
I will just want to comment that another of the parameters that we have in consideration to optimize the sequences are the quantity of pulses that we apply, or not only pulses, also the shape of the gradient. If it is which is
sharp or not,
sharp or not, it is a square, is a sinusoidal, and so it's then the kind of information is totally different that you can extract from them. So there are many parameters there to optimize, also to try to do it in the shortest time.
We have one question from the audience before we move on to the next speaker, from Iliana Ilescu. Any leads for clinical translation?
Yeah, so as I mentioned, we are collaborating with some colleagues that they already have some kind of prototypes to try to implement our sequence, or at least something that is very similar. So in principle, I mean, to implement it in a clinical manner with humans, it should be quite direct. So what is more difficult to know now is what will be for one side, we are developing the methods and the techniques to get this quantitative information. Then the next step is to try to find where will be more useful to study what kind of illness, or to study, I mean, in the clinic, then you have to find what will be more practical and what will be more useful. And this is an exploration that we have to do. At the moment, we will start with our colleague is working with the neurodegenerative diseases. So at the beginning, we will start with volunteers, but in some moment, then we will start to use these quantitative tools to study some diseases and try to find where it works better. But
the focus is on the brain.
The focus will be on the brain at the moment, but in principle, the technique is general, but you have to find the best way where to use it.
Yeah, we expected this last period, this last month, start with that, but well, with the pandemic, it was a bit postponed. But we will do soon.
We're definitely looking forward to seeing more work in that direction. Thank you so much for joining us.
Thank you very much.
Thank you very much.
Okay, let's move on. So our next speaker is João de Almeida Martins from St. Olav's University Hospital in Norway. And he will talk to us about diffusion, relaxation, correlation, MRI.
Hi, let me just start to share screen. So, greetings from the cold north. I hope that every one of you can see my my screen well. And I think there was sort of a mismatch between the exact titles of what Chantal said and what I actually wrote here, but fear not, I will talk about exactly the same thing. Because I'm basically going to talk about my research. I mean, I'm currently in St. Olaf's Hospital, but this is actually relating to the research I performed when I was a PhD student in Lund University in Sweden. So I've been moving more and more north. And it basically relates to how you can extract more information using multidimensional experiments, combining both relaxation with diffusion. So first, to just give a taste of why to use either relaxation or diffusion, let me just give you a very simple intuition about what kind of information do these two measurables give to us. So in terms of relaxation, you can understand it as just a measure of tissue chemistry. You measure relaxation rate if you use an appropriate MRI experiment for that, and the relaxation rate is going to be different depending on what is your composition of the tissue. So if you have just free water, you'll measure a pretty low relaxation rate. If you have water within, let's call it a dirty environment, an environment where you don't have just water molecules, but you have some sugars, you have also some lipids around, and due to some chemical exchange processes, this relaxation rate will be different, will be slightly higher. It will be even higher in an environment where there are even more lipids. So the longer there are the longer are the molecules around, the higher will be this relaxation rate. So you have quite high relaxation rate due to the exchange of protons between the water in this layer. So you can imagine these stacks of myelin layers and some water inside, and the relaxation rate of the water inside those layers will be even higher. And it will be even higher if you don't detect the signal of the water inside those layers, but the water of the myelin lipids themselves, and so forth and so on. Unfortunately, very high relaxation rates will be very hard to measure in an in vivo setting with clinical scanners. So we mostly will be confined to this small region where you have an indication of whether you have free water or water within an intra or an extracellular environment. Much more specific information can be attained with diffusion. And here I will actually build on the same analogy as Gonzalo and Analia in their previous presentation of this way of seeing diffusion of their effects on the drop of ink. So if I drop if I drop a small drop, of fluid on some different cellular arrangements, let's see what are their effects. So let's assume that I have no structure, so it's basically free water, or you can imagine this necrosis, a complete destruction of the underlying tissue. You'll see that this ink droplet will diffuse rather isotropically and rather fast. It'll create like a large blob. If I drop it on a tissue that is defined by small cells with no anisotropic structure, you will see that the diffusion will be restricted. There's some exchange across the membranes, but still it will find a lot of different barriers. So it will be more constrained. And since there is an underlying isotropic architecture around, it will also be isotropic. So we can capture these effects by defining a tensor whose size will tell me something about the underlying media. So the size of my diffusion tensor will tell me about the size, the amount of restrictions it will find in that medium. So if I have a medium with a lot of small cells where the water diffusion will find a lot of barriers, I have a small diffusion tensor, which is isotropic. And if I have a media which I don't have many restrictions, my diffusion tensor will be bigger because, well, it will be diffused faster. And not just the size, but the shape will also tell me something. Because, for instance, if I see here that I have some nerve, some tissue that is anisotropic, the diffusion, the ink will diffuse rather differently, right? I have a faster diffusion along the axis of anisotropy and a much slower diffusion perpendicular to it. So I'll have an anisotropic pattern. And again, I can capture this with sort of a shape of my diffusion tensor. So the shape of my diffusion tensor tells me if I'm in an isotropic media, such as, for instance, these two, or I'm in an isotropic media, such as this one characterized by, for instance, nerve tissue. And mathematically, if I want to describe this, and I will go back and forth with these quantities in my presentation, I can define two measures of the size and the shape. So the size will be basically the trace of my diffusion tensor divided by 3. Well, the shape is basically given by the difference, a scaled difference between the parallel and perpendicular diffusivities of the tensor. Okay, so and with this idea of diffusion and relaxation, I can form an idea of what I expect to find in a voxel. So again, a voxel, we have to keep in mind that an MRI voxel is actually a millimeter-sized quantity. So in theory, will encompass different kinds of tissues. So I can describe it, the collection of microscopic tissue environments within a given voxel by a collection of diffusion tensors and relaxation constants. And they will be different depending on which kind of tissue I have. For instance, here, I only represented my collection of diffusion tensors because I still haven't figured out a way to also represent the relaxation rate in this voxel. So, but for instance, if I have an isotropic tissue that is oriented from the bottom to the top, I'll have a diffusion tensor that is anisotropic and has an orientation that is consistent to that. If I change the orientation, but still keeping the same kind of tissue, we'll see that the diffusion tensors will also change its orientation. And for instance, if I don't have any anisotropy, I have isotropic tissue that is rather dense, I'll have small anisotropic diffusion tensors. While if I have free water, I'll just have large and isotropic diffusion tensors. So when my aim is to actually map all of these quantities into a distribution of different components, where each component corresponds to one microscopic tissue environment within my voxel. So for instance, this is the distribution from these different tissue environments on this same voxel. And I'll have, for instance, these colored points here that have a slightly lower, well, it's not the highest size, not the highest isotropic diffusivity, but it's rather anisotropic. And their orientations are here color-coded by indicated by the color of the points. They correspond to these two anisotropic tissue environments. If, for instance, I go down in a tissue environment that is isotropic and still with a similar size as the one that is anisotropic, and you also see that its relaxation rate, it's still rather high. This corresponds to this one, water inside an isotropic intra-extracellular environment, so water inside isotropic dense tissue. If I focus, for instance, on this population here that has a high isotropic diffusivity, it is isotropic, and it has a low relaxation rate, well, this corresponds to this free water. Okay, and this is then my goal. My goal is to define a recipe to both encode enough information to measure these distributions and then develop some analysis tools to extract the distributions. So we develop, and I will go fairly quickly how this is actually done, but we will define some pulse sequences to acquire and to encode enough information about diffusion and relaxation. They will allow me to acquire a signal in a 5-dimensional space of 5. So this 5-dimensional space means that I have 5 different experimental dimensions that I have full control of, and I can probe independently, somewhat independently, actually. And then I will hopefully transform this very information-rich signal into a distribution of different components. So I will go back to what we can actually do and how we can actually recover these distributions. But for now, let me just spend a couple of slides trying to give an intuition of what we are doing in this step. So a very basic introduction of how you actually would conduct a diffusion relaxation experiment. So you have here, this is our workhorse called a spin echo MRI. Those of you who are not specialists in MRI, do not be scared. It's nothing fancy. What you need to focus is that we have two main different objects that I as an experimentalist can control. I can control these gradient waveforms. They are here color-coded. So these gradient fields, I can control their magnitude, their time trajectory, their orientation. And this is what will encode information about the diffusion processes. I can control the spacing between this first 90-degree pulse and the actual acquisition. This EPI block represents where I actually form the image. So the time between the excitation of the signal and the time that I record the image, I can control. I can increase it or decrease it. And this is what encodes for the transverse relaxation processes. So this is what gives information about relaxation. And what I'm basically doing is that I do a loop where I acquire several images, and each image will be encoded with a given value of B tensors. So these B tensors is the object that I define with these gradient waveforms that will give information about the diffusion process, and relax and echo times, which is the scalar quantity that I control that will give information about the transverse relaxation rate. So what we'll have is a stack of different images with different contrasts, and this contrast will be given by the weighting of relaxation and diffusion. And then just a very small thing. So those of you who are actually familiar with diffusion, MRI, might be finding that these gradient waveforms are maybe a bit too weird. And they are indeed. So this is a flavor that is actually quite the specialty of Wund in a sense. It's called the B-tensor encoding. So this would be maybe what you would be used to see in a diffusion experiment, this Stegel-Tanner design where you have two pulses. And instead you see these where we are firing at the same time at 3 collinear directions and defining a weird time trajectory. This is done to encode more information, but intuitively you can understand this as adding a new extra dimension to your acquisition space, because with these gradient waveforms, I have only control of the orientation and the size of the B tensor. So these B tensors, again, is an experimental object that is defined by the amplitude, orientation, and time trajectory of the gradient waveforms. So I have basically two different dimensions that I can change, its orientation and its size. While if I am a bit more careful and design these non-conventional waveforms, I add a new dimension where I can also change the shape of the B tensor. I can define B tensors that are isotropic and isotropic, planar, stick-like, doesn't matter. And this in the way you can understand it as a way of trying to match the dimensions of my analysis space, because I want to get information about the orientation, shape, and size of the diffusion tensor. So then I match it with the experimental space where I also change the size, shape, and orientation of my B tensor. And then I can define a 5-dimensional space, right? Given by the echo time, which gives information about relaxation, the size, the shape, and the orientation. Well, the orientation is 2 dimensions of my B tensor. And I know that this is very, very brief, but those of you who want to see a bit more details about this B tensor encoding, I highly recommend this recent review, and you can find way more details there and how this actually relates to other processes other than just tissue structure, also about exchange and time-dependent diffusion effects, for instance. So now we just briefly got to see how we acquire a 5-dimensional space, a 5-dimensional signal, sorry, encoded by both relaxation and diffusion. And now let's try to see how we actually transform this into distribution. So the most general way I can describe what I actually measure, how my signal is measured, is as an integral of a probability distribution, that is the object I want to recover, this probability distribution informing me what I have inside my voxel, weighted by 2 factors, by 2 exponential factors that are basically regulating the weighting done by R2, by the relaxation rate, and the weighting by diffusion. And I want to recover then this probability distribution. There are several ways. One of those that I focused the most during my PhD was actually invert this mathematical transform as is, trying to do a direct inversion of the probability distribution. And this relates to basically a Laplace inversion. It's not the most amiable problem, but it's doable. And if we apply this in the in vivo brain, this is actually what we get. So acquiring enough information, I can actually directly invert this relationship. And I have an array of different distributions. So let's just see a couple of results from an in vivo brain. For instance, if I zoom in on a voxel containing only white matter, what I will find is a distribution that has anisotropic components with, a size that is not very high, so indicating that they are not completely free diffusion as expected. The relaxation rates, there's a bit of a dispersion, it's a bit noisy, but they are relatively high. And the orientation, the color of those points will change depending on which fiber population I'm looking at. If I focus, for instance, in a voxel corresponding to cerebrospinal fluid, just free water, what I see is a population, what I extract is a population of components, of microscopic components that have very high diffusivities, very high, the diffusion tensors have a big size, they are isotropic, and the relaxation rates are rather low, as I would expect for free water. Gray matter is perhaps a bit more tricky. It's similar to white matter in some senses, so it has a similar isotropic diffusivity. The overall size of diffusion tensors is similar. The relaxation rates are also on the same order of magnitude. They're maybe slightly lower, but still, compared to this one that I'm seeing it, but similar magnitude. What is different is that they are not as anisotropic. See that here, this ratio between the parallel and the perpendicular eigenvalues is much higher than here. Here, instead, I have this dispersion over isotropic. So I have this sort of butterfly pattern where I have a distribution of different sizes. So I can have microscopic components that are described by like a fat stick, so a chubby stick-like diffusion tensor, all the way up to a planar. Why we have this dispersion is due to two factors. There's a factor of noise creeping in that gives a slight preference to anisotropic configurations over completely isotropic ones. So we cannot for sure say if they are in fact anisotropic, slightly anisotropic, or fully isotropic. So the noise here is a confounding effect. And the dispersion is this butterfly pattern around this because there's virtually undistinguishable signals between, or let's put it this way, like planar diffusion tensors and cigar-like diffusion tensors, they give virtually indistinguishable signals. So it's very, very hard for the algorithm to distinguish them, so it just accepts any of them. But anyway, not as anisotropic as white matter. And if I focus on voxels that contain mixtures of the different tissues, I will find basically what can be roughly described as linear combination of the different individual distributions. For instance, if I focus on white matter and CSF, I see that I have some populations with very high diffusivities. They are isotropic, and some other cluster of points that are highly anisotropic have a given orientation, in this case along X, because I took a point here in the border between the ventricles and the corpus callosum, so it's running from left to right, that have also much higher relaxation rate. And similar situations can be seen for mixtures of white matter and gray matter and CSF. For instance, gray matter and CSF, you really see the separation here, that you have much higher, much bigger diffusion tensors and much smaller diffusion tensors, so higher diffusivity rates and lower diffusivity rates corresponding to CSF and gray matter. But this is, I just spent like 2 minutes just looking at a couple of distributions. This is clearly hard to visualize, and we want to see whole brain. So how can we do this? So there are different ways to condense this information. I will just briefly touch some of them. One of the easiest ones is to subdivide my distribution space into different beams, different regions that I assign where I expect the components to fall. For instance, I can define this thin beam that would sort of capture where I would expect white matter contributions to fall in, because it's capturing microscopic components that are anisotropic. I can define this big beam where I capture all of the contributions with high the isotropic diffusivity rates, and the thick one, basically everything that is the rest, something that should encompass not very anisotropic tissues, that they also don't have very high diffusivities. And if I map this into the brain, I see that I find, well, all of the thin contributions are mostly where I would expect white matter. The big contributions are where I would expect free water, so CSF. And gray matter has this mixture as mostly falling into the thick beam. But here in the deep gray matter structures, I can see these yellowish and orange voxels that tell me that I have a mixture of these two beams. So they have a mixture of both isotropic and anisotropic populations. And if I compare just this signal fractions with an anatomical map derived from a segmentation from a high resolution T1 weighted, I see that they match rather well, at least superficially. And again, it's a bit more tricky. These deep gray matter structures, they don't really fall into a specific bin, but more of a mixture of them. And after I define these bins, I can then select, for instance, different populations and focus on their characteristics. For instance, I can focus on white matter. I can produce different maps of only regions that have white matter, that have these thin contributions. I can see their relative orientations. I can see their relaxation rates or their shape, so the anisotropy of their diffusivities or their size, so how fast do they diffuse. Or I can represent them in another way. I can form in even a more compact representation, and I can use them to derive some glyphs, some ODF glyphs that at the same time indicate crossings and orientation. And the mappings of, for instance, R2 say that, for instance, looking here at this ODF glyph that is colored by the relaxation rate, I can follow the orientations of major fiber pathways. For instance, here the corpus callosum. I can see here crossings between the corticospinal tract and the corpus callosum. You see this sort of X-like ODFs. And I can also see, for instance, populations with different chemical composition. You see here these very red populations that in this deep gray matter population is where I have higher deposition of iron. So they would have a different relaxation rate. And I can distinguish them based on this different relaxation rate. Okay, but this is maybe not enough, because if I focus on these distributions, or if I focus on distribution of white matter, I see that I have like this blob. You know, I don't really can distinguish different components within white matter. But if I look at histology, well, I see different populations. Like I see, for instance, water is seen inside the axons and water outside of the axons. So can we actually distinguish that? Can we access this information? Yes, but we have to take a different analysis approach. We cannot invert directly the signal. We have to build a model. And you know, there's this very famous paper in MRI, in microstructural MRI, that says that every model is a picture. So this is my picture of white matter. I've defined these cylinders where I have an intra-axonal population with its given relaxation diffusion properties, an extra-axonal population with its properties as well, its own relaxation time, its diffusivities, and maybe myelin water. Well, yes, it's there, but I don't really detect it because it decays way too fast for me to detect in typical diffusion experiments. So it will be in practice invisible. All I will see is these two intra and extra axonal populations, and I will model them directly as two different contributions in a model. This was actually the work of a colleague of mine, Björn Mampinen, which showed that you can actually get, if you acquire enough data, if you design optimized 5-dimensional protocols encoding both for diffusion and relaxation using B-tensor encoding, and if you design them carefully, you can very precisely estimate these parameters with minimal constraints, and you can get maps that are anatomically plausible. And this is great. However, there was more recently a problem that we found and we took, which is this relies on nonlinear fitting. So it takes very, very long time to complete the analysis of this kind of data sets, around 8 hours. So as a solution for this, we jumped on the hype train and, why not machine learning? Let's try to fit this with machine learning techniques. So we actually replaced the nonlinear fitting process with a multi-layer perceptron representing our model. And we try to see, this is very unpublished work, this is very recent, and we try to see what we could get, like what are the trade-offs? Can we still get good maps? Can we even train this model because it's very high dimensional? Well, very early results showed that yes, it was quite nice. We get some maps that resemble, so we defined some synthetic data sets, some synthetic maps, and we saw what were their neural network predictions. They agree well visually, and their correlations are also quite good. I mean, they are not excellent, the correlations between the predictions and the targets, but they are good. They are good enough. And more importantly, an 8-hour fitting problem was reduced to just around 10 seconds after the network is trained. So this was quite promising. And with this, I'm already over time. For that, I apologize. So I will conclude my presentation, I can just say, since this is a hackathon, I can say that most of these methods, well, they are mostly implemented in MATLAB. We are MATLAB for life kind of group. And you can find them in publicly available repositories. For the Model 3, I would suggest to look at one that these methods, DTR D2 folder in this one that is my own GitHub repository. For the model base, I would highly recommend you to take a look at Bjorn's GitHub repository. And unfortunately, we haven't still made the machine learning methods available yet publicly, but they will, so keep tuned. And yes, with this, I would like to conclude the presentation. Just give a brief summary. So we talked about methods to encode for both microstructure and chemistry of the tissues using diffusion and relaxation, how to represent them using distributions or parameter maps. And this was not possible without all of my co-authors. So they are cited here. Very many smart people that helped me and made this possible. So, and yeah, thank you, and thank you all for your attention.
Thank you very much, Joao. That was a very nice presentation with pretty images.
Yes.
So if you have a question, you can also, actually, we can enable you to talk. So you can raise your hand and we can enable your microphone. We have a question from Andrada. How stable is the 5D inverse Laplace? Do you use priors?
No, it's unstable. It's noise dependent, but we actually use that instability to find regions of certainty. We use that for error estimation. So we couple it with bootstrapping procedures to sort of get a worst case scenario. We get some variability within our estimates, and we use that to sort of gauge the uncertainty in our estimates. But it is unstable. What there is embedded, you can improve the stability though by putting some priors or adding some regularization criteria, which we are currently not doing, but it is possible.
Any other questions? I just said that I could enable your microphone, and then immediately after I forgot to do that. So apologies, Andrada. I have a quick question. Marco, do you have?
Sorry, there is Paddy who raised the hand.
Yeah. There's so many different places to. I think, Paddy, you should now be able to talk.
Yeah, hi there. Thanks, Joao, for the nice talk. I was just wondering about the machine learning model fitting, and I was wondering whether that's done whether you learn just on voxel-wise signals, or whether you learn on sort of patch-based in a patch-based way.
Currently, this is only voxel-wise, so there's no structural information going into the network. That can also be possible, and I would say that it's an interesting pursuit. But currently, it's done on synthetic data generated from previous. That is generated with some priors related to previous data, to previous in vivo data, and the whole process, the whole training process, is done voxel-wise. So there's no information across voxels. It's effectively not an image that enters into there.
Right, I see. Thanks.
You're welcome.
Matteo.
Yeah, thanks, Joao. It was very interesting. I have a follow-up question on the machine learning part. So since it's 11-dimensional data that I can't even imagine 5, I can't even try to imagine 11. So have you tried to see what happens when you put as an input to the network something that it has not seen during training, and see if it actually fails spectacularly? Because that's what I would be expecting, since it's so high dimensional, and so if it gets it wrong, it should get it very wrong.
Yes, it can fail spectacularly. And that was actually one of the challenges, was to define a training strategy that is both sensible in this generalizability, or well, the span of the space that you that you cover and being accurate. So what we do to remedy that was we try to also introduce some samples, some randomly sampled training samples from the entire parameter space. This reduces our accuracy, but means that the network has seen a bit of points outside of what we would exactly predict to find in vivo. If I use only data from that is compatible with a brain data set, with what I would assume is a healthy brain data set, then yes, then it fails spectacularly. Also, I'm slightly outside of it. So it is still a bit of a point that deserves a bit more optimization from our part to actually design the training strategy, because again, it's 11-dimensional. It's very, very hard to define a sufficiently dense training data set there. So we have to constrain ourselves to a smaller region, but how small that region should be and how to design it is yeah, it's complicated.
Thanks. I really look forward to see the paper and the code, because I'm very curious about this.
Thanks very much, João. We will go on to the next and last speaker of this session, which will be Andrada Janusz from the preclinical MRI lab at the Champollion Foundation in Lisbon in Portugal. And she will also talk to us about microstructural modeling and new data acquisition and modeling strategies for diffusion MRI.
Thank you very much for the introduction, and hello everyone. So I'm gonna share my screen now and the slides. Okay, so I'm Andrada. I've done my PhD and first postdoc at UCL, and I have moved to the preclinical MRI lab at Champollion Foundation in Portugal about a year and a half ago. And thank you very much for the invitation and the opportunity to talk about my work. Marco suggested that I talk about microstructure modeling, but also about multidimensional encoding and time dependence in the diffusion acquisition. So basically, I will give you an overview of current approaches, as well as my experience with diffusion models. And I will cover different projects that I have worked on in the past that cover different aspects of diffusion acquisition and modeling. And then I will have also a brief discussion about modeling the diffusion data. As we have seen in the previous talks as well, diffusion MRI is an indirect, non-invasive probe of tissue microstructure, and it really provides clinically relevant contrast, as you can see here in these stroke images, but also the quantification of microstructural changes, so we can really get more quantitative information about the brain. But also a good visualization of white matter tracts, and this is an example for where diffusion MRI is used to see the tracts around a tumor. So what do we measure with diffusion MRI? As also was presented in the previous talks, we want to learn about the material microstructure from the way the material affects the diffusing water molecules. And this can be described by a probability density that describes the displacement of the water molecules R after a time t. And as you can see here, the standard deviation of the distribution usually increases with time in free diffusion. So what we want to do, we want to measure the average probability distribution in a given voxel. And then we want to use this to infer some information about the tissue microstructure. So let's have a look at how this probability density function looks. And this is also similar to what Joao showed you. So if we look, for example, in CSF, we see that the water can diffuse quite fast. In white matter, we see that when the tracts are more or less parallel, then the diffusion is highly anisotropic. And we can also see more complex diffusion patterns in areas of crossing fibers. While in the gray matter, we have something in between, still slower diffusion and more on the isotropic side. So how do we measure diffusion MRI data? And this is really the standard sequence, which is the single diffusion encoding. It was introduced a while ago in 1965 by Stacy Colin Tanner. And you can see here that we have a pair of diffusion gradients. And usually it is embedded in a spin echo preparation. It's similar to what to the standard sequences that Joao showed as well. But we can also use other types of encoding and readout. So usually what we tune in this sequence is the gradient strength, the gradient duration, the diffusion time, and then the orientation of the gradient. So the simplest diffusion measurement really is to map the apparent diffusion coefficient. So if diffusion is free, then the probability distribution is a Gaussian function. That's why this regime is also called a Gaussian diffusion. This model assumes that we have a single water pool and that the diffusion is unrestricted and isotropic. And in this case, the signal can be written as a mono-exponential decay, where we have here the apparent diffusion coefficient and B, the diffusion weighting, also referred to as the B value, which depends on the sequence parameter. And, then we can obtain maps of apparent diffusion coefficient, and we see that, for example, in CSF, we have really high values of ADC. Of course, this model has limitations because it cannot explain like tissue restriction and deviation from one exponential decay, and also it cannot model the tissue anisotropy. So the obvious step is to have an extension to 3 dimensions and to look at the diffusion tensor. In this case, we can model the probability distribution with a 3D Gaussian function, where in this case the diffusion tensor is a positive definite symmetric 3 by 3 matrix. And then we can estimate the diffusion tensor from at least 7 different measurements, one with non-diffusion weighting, and then with 6 gradient directions. Well, this is like really the minimum requirement, but usually data sets have 20 or more gradient directions. Then from the parameters of the diffusion tensor, we extract usual scalar metrics such as the axial diffusivity, radial diffusivity, mean diffusivity, or fractional anisotropy. And you can see here the example of maps. And these are probably some of the most common metrics used in the diffusion MRI literature, because DTI has been widely employed in clinical practice. You can see here a map of fractional anisotropy, which highlights the white matter tracts and also a color-coded direction of the main fiber orientation. Nevertheless, the diffusion tensor has some pretty big limitations. It cannot account for multiple fiber orientations, and also it cannot account for restricted diffusion. So if we look in the literature, there are really many different approaches to overcome the limitations of DTI. And I want to give you a brief overview and also mention some recent review studies. So we have models which aim to describe the deviation from Gaussian diffusion. These models include, for example, diffusion kurtosis, Q-space imaging, stretch exponentials, and the additional model parameters indirectly reflect some properties of the tissue. Then we have models which account for the complex diffusion pattern in areas with multiple fiber orientations. And these modeling approaches usually require acquisitions with like high angular resolution or HARD. Here we have the diffusion spectrum imaging, Q-ball imaging, which look at the diffusion orientation distribution function, or spherical deconvolution techniques, which look more at the fiber orientation distribution function. And these approaches are important for tractography when we want to look at the white matter tracts throughout the brain and how they connect to different parts of the brain. And then the other approaches are the microstructure tissue models, which assume some properties of the diffusion in the underlying tissue and find a mathematical representation to and fit it to the data. And there are really various model complexities and approaches for different tissue types. The models that have been previously developed for the brain tissue, and here I focus mostly on the microstructural models, look especially at white matter. So the motivation behind the physical models is to explain the effect of the cellular membranes and to extract information regarding cellular features such as density, size, and shape, non-invasively from diffusion MRI data. There are models which assume axons with zero diameters, or we refer to these usually as sticks in the diffusion MRI literature. And there are many such models like ball and stick, NODI, white matter tract integrity, and other similar approaches. Usually they model the intraneurite space with sticks, and then they have one or two other compartments for the extraneurite space, sometimes even a partial voluming with the CSF, for example, in the case of NODI. Then there are also models which aim to map the axon diameter, and you listened to a really nice talk in the first part of the session on mapping the axon diameter. This has started in the 90s with early NMR and MRI models, then with the translation to the clinical and in vivo imaging, and recent developments have been really using the high performance gradients and really high diffusion weighting for axon diameter mapping. There are also models which include the SOMA contributions, and this is something that I will talk about because I'm also involved in these projects. There have been recently a couple of very nice reviews on modeling the diffusion data. Some of them have been previously mentioned in the other talks. So if you're interested in this topic, have a look at them. They're really good. So in the second part of the talk, I really want to tell you a bit about my experience with the diffusion models. And since Marco kindly asked me to cover different topics, I will just give you an overview of the different projects, and I will not go too much into depth like theory or of each project. So first I will talk about the white matter applications for axon diameter mapping with oscillating gradients and extension to multidimensional encoding. Then I will talk about my more recent work on modeling gray matter microstructure with Sandy, as well as simulations that look into effect of the branching of the dendrites. So one of the questions that we were looking at during my PhD work was whether we can improve the estimation of axon diameters if we use other sequences than the standard single diffusion encoding. And which sequences should we use given the hardware and time constraints of the acquisition? So, we have performed various studies that included automated optimization, analytical models, and really sensitivity analysis over a wide parameter space of the SD and oscillating gradient sequences. And basically our results showed that indeed replacing this pulse gradient with an oscillating gradient can improve our estimates. And this is also related to the previous talk from this series, that they have also used non-conventional sequences. So the results showed that basically high frequency oscillating gradients can be very useful for estimating the intrinsic diffusivity parameters, while the low frequency oscillating gradients can be used for mapping the small axon diameters in case of unknown fiber orientation and dispersion, which is really the case throughout the brain. What is the best protocol? Probably a combination of single diffusion encoding, high and low frequency OGSEs, but then we really need to have like numerical optimization for each problem specifically. For example, if we look at the central nervous system where the axons are smaller, or if we look at the peripheral nervous system where we have larger axons. So this work also prompted a resolution limit about the smallest axon diameter, which can be distinguished from 0, which depends on the gradient strength and SNR. And since the majority of axons in the brain are quite small, basically we see that the sensitivity to the larger, we have sensitivity to the larger axons from the tail of the distribution. And more recent works really look at high gradients and high diffusion weighting to estimate axon diameter. The previous techniques I was telling you about usually use a collection of diffusion sequences with a given gradient orientation per measurement. And in the previous talk, Joao also referred to this as linear encoding. But such sequences have a limited sensitivity to the complex configuration of pores. If we take as an example this substrate of randomly oriented pores, and isotropic pores with a size distribution, they will have a pretty similar response in the linear encoding. So sequences with varying gradient orientation, and here we can talk about double diffusion encoding or the B-tensor encoding and Q-space trajectory encoding that Joao explained so nicely in the previous talk. So such approaches really disentangle between the more complex substrates, and really they can improve the measurement sensitivity and specificity to the microscopic tissue anisotropy, irrespective of the orientation distribution. And my work has focused on this class of sequences that we call double diffusion encoding, where basically we put together two pairs of diffusion gradients, and we can vary their orientation independently. So usually in DD studies, the microscopic anisotropy is estimated from measurements with parallel and orthogonal gradient orientations at a given B value. And you can see here that this is an effect on the order of B squared. But we have done lots of experimental work on this, and we have seen actually a really pronounced B value dependence when we estimated the microscopic anisotropy. You can see here that it really varies a lot between B1000 and B4000. And this is actually due to higher order terms, ignoring other terms when calculating the powder average, as you can see in this expression. So what we proposed is to actually do a very simple correction and include the terms in the order of B to the 3. And in this case, we see that in simulations as well, the corrected microscopic anisotropy really overlaps with the ground truth values in this system of randomly oriented pores, while the single shell microscopic anisotropy metrics really decreases with the B value. So to further probe the tissue microstructure, we can add more degrees of freedom in our measurements. So we can also vary the diffusion time or replace the gradient pulses with oscillating gradients. And this is what we proposed to do to look at the tissue microstructure. And the link between the underlying tissue microstructure and the time and frequency dependence of the microscopic anisotropy, I think is still an open question, but our preliminary results show that restricted diffusion along the fiber or a restricted diffusion inside the spheres or exchange can be some of the factors that explain the time and frequency dependence of microFA that we saw in the mouse. So next, I would like to change a bit the gear and look at modeling the gray matter microstructure. And this is a project that I have worked with together with Marco Colombo, one of the organizers of the hackathon. So most common models of brain tissue microstructure, they account for the intraneurite space, which usually are modeled as sticks, and then the extraneurite space. And what we proposed was actually to include the signal coming from the soma. So now we have the intraneurite space, the intrasoma space, and the extracellular. And when we looked at this model, we can see that indeed it can explain really well the signal decay that we see both in white matter and in gray matter. Of course, the model parameters are very different in the two regions. So then we have applied this model, and we looked at the model parameters both at like in vivo human data, and we see that the map of soma fraction looks very similar to the Nissl staining in the human brain. And these are some really very recent results from from the preclinical MRI lab at Champalimaud, where I have been working on using these methods for in vivo mouse imaging. And you can see that these maps indeed reflect the area where we expect to see high soma fraction, and here high neurite fraction respectively. So from these results, we can see that we can estimate the apparent soma density with the Sandy approach. But what about other cellular features? Can we use diffusion MRI to estimate cellular branching? And this is the question that I have been investigating in a recent simulation study, where we used Monte Carlo simulations with 3D meshes that look like this, that have connected somas and neural projections, and then we can change the branching order of the neural projections as well as the soma size. And, we have simulated the diffusion signal for a single diffusion encoding over a wide range of p-values and diffusion times, as well as for double diffusion encoding over a wide range of mixing times. And while the single diffusion encoding indeed provides sensitivity to soma size as we expected, we actually showed that the double diffusion encoding and its mixing time dependence can really distinguish between cells with different branching orders, while this cannot be done with the single diffusion sequences that we have investigated. So in terms of modeling diffusion MRI data, it is really a very vast landscape, because there is a large variety of techniques to model the diffusion MRI signal for different applications. And different techniques have different modeling assumptions, especially for biophysical models. Some account for permeability, some don't. Some account for Gaussian diffusion or non-Gaussian diffusion in the extracellular space and model additional effects. So it's really important to understand what model we're applying. In general, multi-compartment models are computationally complex if estimated using non-linear optimization. Can also be degenerate, meaning that different combinations of parameters yield very similar signal values. Then to solve some of these issues, we can use fitting constraints or as well as prior or fixed parameter values. And here we also need to be careful if these values make sense, and how we can validate or how can we understand these values. And research studies need to really explicitly describe these aspects of their modeling work in the method section. Also, the estimated parameter values might depend on the quality, the type, the amount of the acquired data, if it has multiple diffusion times or not, what is the maximum p-value, and so on. And as well as the sequence parameters that have been used, for example, the gradient duration, the diffusion time. So in general, I think for biophysical models, parameters need to be interpreted with care, especially in the context of applying them to characterize a certain disease. So which model do I choose? Well, it really depends on the application and on the acquisition strategies and the model assumptions that we are making. Some current and future trends, I think people are looking into better modeling to make the models more accurate and look at axonal undulation, permeability, thickness of the myelin sheet, diffusion in the extracellular space, and so on. Then analytical models, incorporating all these features are intractable computationally. So machine learning approaches gain popularity, as you have seen in the previous talk as well. We can also do like dictionary learning and machine learning for faster parameter fitting. What we can improve is the acquisition as well. We can use oscillating gradients. We can also select the acquisition and target it towards the parameter, towards the tissue type that we're interested in, as you have seen in the first talk today. We can vary the gradient orientation and look at multidimensional diffusion, and we can also include relaxation measures. So I think there is scope for really improving our acquisition and our modeling, and I'm really excited to see that there is so much really cool work going on this topic, including all the projects that have been proposed in this hackathon. Yeah, thank you very much for your attention. Thank you to the preclinical MRI lab at Champalimaud and Dr. Noam Shemesh, the head of the lab, but also my previous lab at UCL, with whom I've been working on some of the projects that I showed you in this talk, especially Marco Palombo, Danny Alexander, Ivana Drobniak, and the other colleagues. And of course, again, thank you for your attention.
Thank you very much, Andrada, for that great overview and your new exciting work. That looks very promising with these new new contrasts that you can show with these sequences. I will try to keep an eye on all the channels that questions can come in. So please post your questions in the Q&A on YouTube. Raise your hand to ask your questions. In the meantime, I have a question. So these oscillating gradients, they are very exciting. What are your thoughts about translating this to in vivo human imaging, because you have so much limits on gradient strength and slew rate?
Yes, so I think the main issue with the gradients, oscillating gradients on the human scanners is the overall lower B value compared to the single diffusion encoding, and also limitations, especially with respect to like peripheral nerve stimulation. But I have seen some like really encouraging results with the low frequency oscillating gradients. So I think it's getting there. So I really see the potential of translating these sequences with care and with tests, but I'm optimistic.
So you think even low frequency gradients would already provide us with new information?
I think so, especially if we're looking at like axon diameter, for example. They can really complement the information that we get from the single diffusion encoding, the standard.
Yeah. Any other questions? So you also work with the animal scanner at quite high field. So how do you see that trade-off between high field and strong gradients?
Okay, so the high field, obviously it gives you a lot more SNR, but also lower T2 values, right? So that means that we're more restricted in terms of what echo times we can use in our acquisition. So I have been working a lot ex vivo on a scanner, which is a 16.4 Tesla with gradients up to almost 3 Tesla per meter. So really, really strong gradients. But then, the echo time is a bit lower. So we can use usually, 30, 40 milliseconds echo time. So there is this trade-off. But indeed, working on a preclinical, scanner gives you more degrees of freedom. And plus we can, easily implement our own sequences, which for clinical scanners is a bit more difficult. You need approval, research keys, and things like that. So we have more flexibility and higher gradients, higher field, but there is always this trade-off between relaxation and higher field.
Yeah, definitely. I'm curious to see where it all goes, where we are in 10 years with our hardware.
Yeah, me too. I'm really curious to see where we're going. Definitely, I think more machine learning, that's for sure. I think it already started pretty fast.
Yeah, definitely. All right, if there are no further questions, unless I missed some, then we can conclude the session. And thanks all the speakers again for their interesting talks and discussions. So the program gives us a bit less than half an hour break. So we start again at 4 p.m. UTC. So Matteo has just posted a link to the hacking space. So please go there. You can go to your breakout rooms. If you haven't been able to attend yesterday, there's still plenty of opportunity to join a project. So please join us and let us know if you have any problems joining a project. And I'm just looking at the other organizers if I'm forgetting anything. No? Okay. Well, then I hope to see you all after a good coffee and tea break in 20 minutes.
Thanks, everyone.
Thank you all for joining us.
Thank you very much.
Thank you.
Thank you very much.
Yeah, thank
Thank
you.
Thank you.
Shall I stop the YouTube too?